Hello, grade 7 students. I'm teacher Gwen, and today we're going to explore something amazing that's happening inside your body, cell division. But before we proceed, let me ask you a question first. Have you ever wondered how your body grows, heals, or even how life begins? It all starts with tiny cells working behind the scenes. Every living thing from the smallest insects to the tallest tree is made up of cells. These cells don't just stay the same. They divide, multiply, and change to help the body grow, repair itself, and create new life. So in this lesson, you'll learn about cell division, the process that allows one cell to become two or more. You'll explore two types of cell division, mitosis and meiosis, and learn how they help living organisms grow and reproduce. By the end, you'll understand how your body keeps building itself and how new life begins from just a single cell. So what is cell division? Cell division is an important biological process where one parent cell splits to create two or more new cells called daughter cells. There are two main types of cell division, mitosis and meiosis. Each one has a special job in your body. Mitosis helps you grow and heal. For example, when you get taller or recover from a cut, that's mitosis at work. Meiosis, on the other hand, helps with reproduction. It creates special cells called sperm and egg cells, which are needed to start a new life. Do you think cells suddenly divide, or do they follow a process? So for your knowledge, cells do not divide instantly and randomly, because before a cell divides, it goes through a series of steps called the cell cycle. This cycle has two main parts, interphase and the mitotic phase or M phase. Let's break down the three subphases of interphase. In G1 phase, the cells grow bigger and prepare to divide. It builds up the needed materials and energy for the next steps. The next phase is the S phase, where the cell copies all of its DNA. The S stands for synthesis, meaning making or building. This phase ensures that each new cell will have a complete set of genetic instructions. And the last phase is G2 phase, where the cells checks the copied DNA and begin organizing and condensing the genetic material. It makes sure everything is correct and all prepares all the things needed for division, including a full extra set of chromosomes. After interphase, the cell enters the M phase, where it divides through either mitosis or meiosis. Now, be ready as we're about to explore the two types of cell division. Let's talk about mitosis first. Mitosis creates two identical daughter cells. These cells have the same number of chromosomes as the parent cell. Mitosis helps your body grow and repair itself. It includes several stages. The first one is prophase. This phase occupies over half of mitosis. The cell prepares for cell division by copying the DNA, and the nuclear membrane and nucleolus are starting to break down in this process. Chromatin condenses into chromosomes. A centromere binds the double-stranded chromosomes together at one point. Spindle fibers begin to form, which are microscopic protein structures that help divide the genetic material in the cell. The second one is metaphase. At the equator of the cell known as metaphase plate, the now double-stranded chromosomes called chromatids align themselves. Each chromatid is connected to the spindle by a structure called kinetochore in the centromere. The third is anaphase. This is the shortest stage. The chromatids are pushed by forces originating from the poles to pass. Consequently, the centromere divides into new, single-stranded chromosomes splitting the chromatids. After that, the chromosomes travel towards their respective poles. The fourth one is telophase. During this stage, the opposite poles have now reached the chromosomes, and the spindle fibers disappear. The chromosomes uncoil at the poles, and the nucleus and nuclear membrane start reforming. But the cell is not yet completely separated. To complete the cell division after stages of mitosis, a process called cytokinesis takes place. In this process, the cytoplasm simultaneously divides, and the cell is separated into two by a plasma membrane. The formation of the new plasma membrane completely divides the cell, forming two identical daughter cells. But did you know that cytokinesis happens differently in animal and plant cells? In animal cells, the creation of a cleavage furrow is necessary for cytokinesis, where the cytoplasm divides and the parent cell becomes two daughter cells. It pinches the cell off or splits it into two. In plant cells, cells do not pinch off. Instead, between the two nuclei, a new structure called the cell plate forms to develop into a new cell wall. Let us now proceed to the other types of cell division, which is the meiosis. Meiosis is a bit different. It creates four daughter cells, each with half the number of chromosomes. These cells are used for reproduction. Meiosis happens in two parts, meiosis 1 and meiosis 2. Let's go through meiosis 1. It is the first round of cell division in meiosis. This stage starts after DNA replication and reduces the number of chromosomes by half, meaning it changes one diploid cell or 2N that contains two sets of chromosomes into two haploid cells, each containing only one set of chromosomes or N. It includes four stages such as the prophase 1, metaphase 1, anaphase 1, and telophase 1. In prophase 1, the chromosomes have already been duplicated. Each chromosome is made up of two sister chromatids joined at the centromere. The homologous chromosomes, or one from each parent, pair up in a process called synapsis, forming structures called tetrads or four chromatids in total, with two from each homologous chromosomes. While in this position, the process known crossing over occurs, where genetic material is exchanged between homologous chromosomes, increasing genetic variation. Metaphase 1. The paired chromosomes known as tetrad line up in the middle of the cell called metaphase plate. Their arrangement is random, which also adds to genetic diversity. Anaphase 1. The homologous chromosome pairs separate and move to opposite sides of the cell. The direction each chromosome moves is random and doesn't depend on its partner. Telophase 1. The chromosomes reach the opposite poles. A new nuclear membrane forms around each group. Then cytokinesis takes place, dividing the cytoplasm and forming two haploid daughter cells. Each cell now contains one chromosome from each homologous pair, meaning it has half the original chromosome number. Let us now move forward to the second round of cell division meiosis known as meiosis 2. Meiosis 2 is the second round of cell division in meiosis. In this round, there is no DNA replication involved. This process is similar to mitosis, where the separation of the sister chromatid occurs. However, this process starts with two haploid cells that are formed in meiosis 1. These cells divide, forming four haploid daughter cells. Each cell contains one complete set of chromosomes. This also includes several stages. Prophase 2. The prophase 2 begins after telophase and cytokinesis. The sister chromatids remain attached at the centromere, where the nuclear membrane disappears and spindle fibers form, and chromosomes prepare to divide again. Metaphase 2. During this stage, the chromosomes line up individually along the center of the cell, called the metaphase plate. Unlike meiosis 1, they're not paired up. Anaphase 2. In this stage, the sister chromatids are pulled apart and moved to opposite sides of the cell. This stage ensures that each new cell gets the correct number of chromosomes. Telophase 2. This is the final stage of meiosis 2, where chromatids reach the poles and a new nuclear envelope forms around each set. Then, cytokinesis completes the process, dividing the cytoplasm. Now we have a total of 4 haploid cells, each with single-stranded chromosomes. Each of these cells is unique. That's why siblings can look different from each other. When a sperm and an egg join, they form a zygote, a cell with a full set of chromosomes that grows into a new organism. Let's compare mitosis and meiosis. The purpose of mitosis is growth and repair, while meiosis is for reproduction. The number of daughter cells of mitosis is 2, while the meiosis has 4. The chromosome count of mitosis same as parent or diploid, while the meiosis is half of parent or haploid. The genetic identity of mitosis is identical, while meiosis is unique. Mitosis helps your body grow and heal. Meiosis helps create new life. Both are super important for living things. Let's remember why cell division matters. It helps us grow, repair our bodies, and reproduce. Without cell division, life wouldn't continue. It's one of the most important processes in biology. Today, we learned that cells divide through mitosis and meiosis. Mitosis creates identical cells for growth and healing. Meiosis creates special cells for reproduction. Both are part of the cell cycle, and both are essential for life. Here's a quick recap. Cell division helps with growth, repair, and reproduction. Mitosis creates 2 identical cells. Meiosis creates 4 unique cells. The cell cycle prepares cells for cell divisions. Thanks for learning with me today. Keep exploring, keep asking questions, and remember, science helps us understand how life works one cell at a time.